Welcome back. For our next video, this will be video number 5. We will be finishing up cellular respiration. We'll be finishing up chemiosmosis. We'll talk about electron affinity, why we need to breathe oxygen. This is called cellular respiration after all. We'll look at glycolysis and Krebs cycle right here, and then we'll also look at anaerobic versus aerobic respiration. And of course, eventually we have to address the equation itself. And don't forget, once this video is done, this is video 5, there are 2 free response that you'll want to look at. On our Canvas page, once again, out to the side where it says files, there's a file that says AP, if you click on files, AP Bio FR referenced in YouTube vids. And on there, if you scroll through it, you'll eventually get to 2019 number 3. Again, the yellow call out box will tell me I should be doing this after review video 3. And then also 2015 number 2. Again, the yellow call out box will tell me that I should be working on that after this video. And then we'll talk about that in one of the following Zoom meetings. Okay. So we left off and we were talking about how Atp Synthase is hollow, it spins. The reason it's hollow is so that protons, these are little pluses that flow through. When protons flow from high to low concentration down their electrochemical gradient, then this will spin. As ATP synthase spins, it synthesizes ATP by joining ADP and phosphate, like that. Well, how do I make sure that protons are always flowing? They flow from high to low because proton pumps pump them into the inner membrane space. How do I power those proton pumps? With electrons. So I have these 2 electron carriers, NADH when it's full, and FADH2 when it's full. By full, I mean full of electrons. They will drop off their electrons at the electron transport chain. So NADH drops off its electrons and it powers 1, 2, 3 pumps. It drops off energy here, here, and here. FADH2, the electrons are not carrying as much energy. So they actually only eventually power pump number 2 and pump number 3. So a molecule of NADH actually provides more power than FADH2. If you want to think of it back to energy levels, if I have electrons being carried at this energy level versus electrons being carried at this energy level, this one has more potential energy. Right? The further I have to fall, the more potential energy I have. If this guy falls, a foot, and this guy falls 2 feet, he has more potential energy. I can hear the difference in sound. I don't know if you can on this video, but that versus that. So that's essentially what's happening here. These are higher energy electrons than these, so these can power pump 2 and 3. Okay, so electrons are dropped off by these electron carriers. Once NADH drops off his electrons, now he's empty-handed, and he'll be called NAD+. Once FADH2 drops off electrons, it'll be called FAD, no pluses. Okay, so the electrons go from pump to pump to pump. Great. Well, by the time a few electrons go from pump to pump to pump, and then we drop off more and more and more, eventually electrons will start to pile up on this last pump. So imagine these 3 expo pins are pumps. And if I go and I take some electrons and I send them to pump number 1, who uses some energy, sends it to pump 2, who uses some energy, sends it to 3, who uses energy, now pump number 3 is holding those electrons. Well, if pump number 3 was completely neutral, now pump number 3 has negative charges, right? I don't know that pump number 3 was 100% neutral, but it's getting more and more negative is my point. So then we steal some more electrons. Thanks to NADH and FADH2, they're delivered. They power pump 1, power pump 2. At some point, I can only pile so many electrons onto that 3rd pump before it can't take any more electrons. At some point, I'm so negative that when I try to put electrons on there, they're repelling each other. So eventually, pump number 2 can't hand off any more electrons to pump number 3. So now those electrons get piled up at pump number 2. Okay, fine. And I don't know how many it can hold. I think it's just a couple, but don't quote me on that. Pump number 1, I drop off some electrons. Okay, so eventually I'm NADH, or I'm NAD+, Now I'm NADH. NADH is actually holding 2 electrons that it can donate. I do know that is true. And so now I'm NADH again, NAD+. Now I'm NADH. I can't drop off any more electrons. So nobody's getting any new electrons because that guy is full, that guy is full, that guy is full. Their hands are full of electrons, so to speak. So I can't drop off any more. So now they're getting no new electrons. They're getting no new energy. So now they have no new power to pump protons into that intermembrane space. If there's no protons in there, they're not flowing through ATP synthase to make it spin to make ATP. So now I'm going to die. But just like last video, I don't wanna. So what I need to do is find a way to get rid of these electrons. If I can have something else come in here and get rid of these electrons, oops, that was fatal. Something takes away those electrons, then now these electrons can move over, these can move over, and NADH can come in and drop off more. If somebody will come in and steal away those electrons, these guys can move over, these guys can move over, and then another FAD can come in and drop off his electrons as these guys move over. FAD could go to pump 2. Technically it's that protein right before pump 2, but power pump 2 and pump 3. So I need somebody that takes away electrons. So think back to review video 1, who's great at hogging electrons? Who was the second greatest electron hog on the periodic table? Oxygen. So this is why we need oxygen. This is why we call this process cellular respiration. Cellular respiration. If I ask my son, what does respiration mean? He'll know breathing. If I ask him, what gas are you trying to breathe in? He'll say oxygen. What gas are you trying to breathe out? Carbon dioxide. So respiration is breathing in oxygen and getting rid of carbon dioxide. Why do we need to breathe in oxygen? Because that takes away our used, our spent electrons. So as the electrons go from NADH to pump 1, to pump 2, to pump 3, then oxygen will come in. The oxygen in the air that we breathe is O2, not just regular old O. From chemistry, you might remember Brinkelhoff, all the diatomic ions. And then oxygen is converted into water once it picks up electrons. So I would have an O2 would eventually end up making 2 waters. I'll explain that process in just a second. You're like, wait, where did H come from? You can't create or destroy matter. So where did the H come from? We'll get there in just a second. So oxygen's coming in and grabbing electrons and taking them away. So as long as I breathe oxygen, then I can keep stealing electrons from glycolysis and Krebs, food, and they will power these pumps, which means this machine will keep running. If I don't have oxygen, then everything backs up and I die. For example, if I consume cyanide, remember from class, there was somebody that was putting cyanide into Tylenol and putting it back on the shelf somewhere in the early 80s. It might have even been 1980. I think it was like 81, 82 though. But you could Google cyanide Tylenol killer. And basically people would take their Tylenol, and the Tylenol had been replaced with cyanide powder instead. And so because of that, the safety seal was invented. Well, how is it that cyanide kills me? When we used to execute people, like capital punishment, in the gas chamber, they were killed with cyanide gas. So how is it that cyanide kills me? Well, it turns out that cyanide binds to this last pump, and it blocks oxygen from taking electrons away. So in a way, you're suffocating, right? Because that means that now the electrons pile up. They pile up here, they pile up here. Now NADH can't drop off its electrons, so now the Krebs cycle stops. Everything backs up. If step number 72 in a moving assembly line stops, then step 71, 70, all the way back, backs up too. So that's why you don't want to take cyanide. Okay. So the next thing that we want to look at is electron affinity. Electron affinity and electronegativity represent the same idea. Technically, electronegativity is within a molecule. So electronegativity was our example from review 1 was water. Oxygen, within a single water molecule, oxygen is hogging electrons more than hydrogen because it has a higher electronegativity. Now, if I go between or if I go between two different molecules, molecule A and molecule B, if molecule B will pull electrons from A, it has a higher electron affinity. Affinity means to like. I have an affinity for my wife. So if we're between molecules, it's technically called electron affinity. On the AP Bio test, if you said electronegativity, it'd probably be okay. But here's what I'm getting at. NADH has a certain electron affinity. I don't know how much. I'm just going to make up a number. 1. This guy is 1.5. This pump is 2. This is 2.5. And then oxygen is even higher than that. So the electrons go from him, to her. I'm not sexist. To her, to her. So that's why they go that way. It's almost like you have the guy, the stronger guy, the stronger guy, the stronger guy. And you give a treat to the first guy, it gets stolen from him, gets stolen from him, gets stolen from him, because they have a stronger attraction, so to speak. So electron affinity increases as we go from NADs and FADs to the pumps, to finally oxygen has a really high electron affinity, which makes sense. Okay, so now how does O2 turn into water? This part is unlikely to be on the AP Bio test, but I think it explains a lot and it closes the loop, so it gives you a general better understanding. But I don't think they will directly ask about this step. So here's how it works though. This will tie in with photosynthesis and it just sort of like completes the whole picture. So over here, I've got oxygen. Actually, let me erase a little bit of Christe here. So what's happening right here in this orange reaction is this. I've got oxygen. On my pretend periodic table up there, oxygen is in column 6. So that means oxygen has 6 valence electrons. Remember, valence electrons are the electrons on the outermost shell. So those are the ones that determine a chemical's reactivity. Just like my shirt. If I was wearing 3 layers of shirts, electron layer 1 and 2 and 3. The shirt on the outside is the one that reacts with the environment. Like if I bump into a wall that's got fresh paint on it, which shirt is the one that is affected? The outer one. So the outer shell of electrons is the one that determines chemical reactivity because that's the one that bumps into stuff. So here's chemical A, here's chemical B. The outer shells are what interact with each other, not the inner shells. That's why valence layers get a special vocab name, valence. So here's an oxygen, and here's an oxygen. These are the valence electrons. Try to make them big enough to where you can see them. And so right now, oxygen is neutral. Okay. If I look at the periodic table, right there, oxygen is neutral. It would have a grand total of, so it's got two electrons in its first layer and then 6 in its outer layer, and it has 8 protons. So oxygen looks like this on a periodic table. If I was drawing this for oxygen, it'd have an 8 and a 16. So it has 8 protons and 8 electrons. But the valence shell only has 6. So right now, this guy has 8 protons, 8 electrons. He's neutral, but he's not stable. Neutral and stable are not the same thing. So because he has 6, he's going to seek, again, I know he doesn't have wants and needs, but he's going to seek stability by gaining 2 more electrons to complete his octet. And so he'll form covalent bond. That's why O2 exists in nature. O on its own would immediately bond with something around it to become stable, maybe a few hydrogens and become water, or maybe another oxygen and become O2. So I bring in O2. O2 will attract electrons, so that O2 is this O2. I'm going to attract these green electrons that I'm passing along. So what will happen is this guy right here, that proton, and it will grab electrons coming from NAD or FAD. That proton will grab an electron right there. And this proton will grab an electron right there. Well, now oxygen has 2, 4, 6, 8, 10 electrons. Same thing over here. grabs 2 electrons. So now it has too many electrons. So if those green electrons stay, then this bond goes away. So let me go ahead and make those circles. Okay, now, this oxygen still has 8 protons, but it just gained 2 electrons, didn't it? Right, so it's got 2 on its inner layer, plus it has 8 out here now, so now it's got a grand total of 10 electrons, but it's still got its 8 protons. Same thing with this guy. Okay, so if I have 8 protons and 10 electrons, I've got a 2 negative charge. So does this guy. Okay, 2 negative charge. What would I be attracted to, positives or negatives? Positives. What's flying all around me? What's like circulating in here and then going back through ATP synthase over and over again? That's right, a bunch of positives. So, because of this, I'll now attract a positive. Positive, positive. Okay, what is a proton and an electron together called? Hydrogen. So that green and that red just made a hydrogen. That green and that red just made a hydrogen. So now I have an oxygen with a hydrogen and a hydrogen, known as water. I have an oxygen. I have an electron. I have a proton. An electron and a proton together is called a hydrogen. There again. So I have 2 water. So for every O2, I will pick up 4 new electrons, and then I'll pick up 4 protons to make 2 waters. So if I go and I look at this chemical equation, I'm going to take glucose, I'm going to steal electrons from it, oxygen is going to take those electrons away, and then one of my products will be water from right there. Okay, now let's go back to the very, very beginning and we'll run through this all at once. Now here, from glucose to pyruvate, there's these 10 reactions. Where do they happen? Does it look like they happen inside or outside of the mitochondria? Outside. What do we call those? We're splitting glucose, so it's glycolysis. Again, lysis means to split. So essentially, if I take a glucose right here, and glycolysis, again, glyco means sugar, lysis means to split. If I have a glucose, glycolysis is 10 reactions, and I basically like tweak this. So I like tweak it, tweak it, tweak it, tweak it. It rips in half. In the end, how many pieces do I have? 2. And they're identical. So those pieces are called pyruvate, and I'll have 2 of them. You might also have pyruvic acid. Again, we saw this earlier. When something ends in ate or an ic acid, that just means it's gaining or losing its proton. So pyruvic acid has its hydrogen ion. Pyruvate means it's given it away. But they're essentially the same molecule. Okay, so in glycolysis, I'll end up inputting a couple of ATP, like 2 ATP. But in the process, I output 4 ATP. They call this substrate level phosphorylation. Down here, when I'm making ATP by using proton pumps and ATP synthase and spinning all this cool stuff, they call that chemiosmosis. To me, I've read how the name came to be, but to me the name is not that helpful because osmosis is like water diffusing, and it's not water diffusing. So that's chemiosmosis. The way that we make ATP up here is just regular old, like an enzyme, so they call it substrate level phosphorylation. I take my substrate, ADP, and I phosphorylate it. I add a phosphate, ATP. So they call it substrate level phosphorylation. Why the level? I don't know. I would just call it substrate phosphorylation, but they didn't ask me. So if I have glycolysis and I don't have any mitochondria, I can still net 2 ATP. So this is one of the ways that maybe before mitochondria came into being, things could live, even though they couldn't make their own ATP. In modern times, bacteria don't have mitochondria. They can still get a couple of ATP from glycolysis. They also do kind of a less efficient version of chemiosmosis as well. But even before mitochondria come to be, maybe this is how things existed. So we're still refining that hypothesis, but we think that glycolysis was first. That's what we think today. Okay, so I when I go from glucose to pyruvate, again, I started here and I broke it off. I'm only making a couple of ATP. Down here I make like 30-ish ATP. Your teacher at the end of this equation might say 30 ATP or 36 or 38. I put 30 plus. So depends on the book. At maximum efficiency, we might make 36 or 38. By the time we ship everything around and we're kind of more realistic, a lot of biochem books will say, yeah, it's more like 30. But the point is this, we make like 2 up here and we make 30-ish down here. So this is where we make most of our ATP. So in glycolysis, what we really want is we want to steal electrons. So I steal electrons. That helps me eventually make some ATP. When I'm done and I've got pyruvate, I want to send each pyruvate into the Krebs cycle so that I can produce a bunch of electron carriers. Here, I'm producing 1 electron carrier. And since it's after I've split, I technically produce 2. Like I get 1 electron carrier out of here and 1 electron carrier out of here. But whenever I go through this process, I get 3 NADHs and 1 FADH for every pyruvate. So I'll take each pyruvate, send it through there, get 3 NADs and an FAD. I'll take the next pyruvate, send it through there, 3 NADs and an FAD. So what's the point of the Krebs cycle if I'm just kind of spinning around in circles? The point is I'm stealing electrons to power this. So the main idea of glycolysis and Krebs is make a few ATP, but mainly I need to strip off electrons to power these pumps. So what I really want out of food is electrons. So I think of it like this. Glucose is C6H12O6. So pretend that this is a carbon. This is a covalent bond, shared pairs of electrons, and I bond that to another carbon. I'm making C6H12O6. I'm just following the carbons. I'm not following the hose. It's a good lesson. So I've got 1, 2, 3, 4, 5, 6. I guess that guy technically doesn't need a bond on the end. So what I do is I go through glycolysis, and in the process, I break off. I break this down because I'm stealing electrons. What was holding all these carbons together? Covalent bonds. What are covalent bonds made out of? Electrons. But I'm stealing electrons, right? So NAD can steal electrons and send them to the electron transport chain to power. Okay, now, I've got these 2 pyruvates. Are there still pen caps, chemical bonds, electrons in there? Yeah. So I'll send this pyruvate through the Krebs cycle. And in the process right here, technically I give off one of my carbons as a CO2. I breathe it out. I send the rest of it through, and as I'm stealing electrons, to power these pumps, the electrons were the bonds holding all these carbons together. So now I just have these loose carbons. I don't need the carbons. So again, I breathe them out. That's 3 I've breathed out so far. I need to breathe out a total of 6. Where do the other 3 come from? The other pyruvate. So again, as I send this out, breathe out the CO2. I steal more bonds, more electrons with NAD and FADH2. Those electrons go and power these pumps. Now I have these extra CO2s, I breathe them out. So the CO2 that I breathe out came from the food, the glucose that I ate earlier. So right now, I'm breathing out CO2s from like breakfast or whatever. If I don't eat for like 2 days, then now I'm breathing out CO2 from whatever I'm eating. But if I haven't eaten food in 2 days, what am I eating? I'm eating myself. And as I get rid of CO2, I'm losing weight. So if I don't eat for 2 days, I'm going to keep breathing out CO2. And after 2 days, I will weigh less on the scale, control for water weight and stuff, but I will weigh less. And I will have less body fat and less muscle on me because I breathed it out. I broke down muscle and fat and sugar to survive, and I breathed it out. That's why I lose weight. So I breathe out CO2 there. I pull off some electrons, I get rid of some CO2s, and then I get a measly 1 ATP per turn of the Krebs cycle. I have 2 turns because I have 2 pyruvates. Okay, so let's walk through the whole thing one more time. If you've got it, you can fast forward a little bit. Okay, glycolysis happens outside the mitochondria. That matters because it can happen without mitochondria. In the process, I make net of 2 ATP. That's important. And I end up with 2 pyruvates. Along the way, I steal a few electrons with NAD. I send those electrons to this pump. I take the 2 pyruvates. There's still a whole lot of electrons in there, so I send them into the Krebs cycle. In the Krebs cycle, I steal a lot more electrons. Those electrons will go and power these pumps. The electrons go from pump, I'll just call it pump 1 to 2 to 3. They have other names, but complex 1 or 2 or whatever they're called. I go from 1 to 2 to 3 because that guy has electron affinity, that one it's higher, or that one has an electron affinity, that one it's higher, that one it's higher, and then they eventually end up on oxygen. Oxygen is the final electron acceptor, is kind of the words that we use. It's the final electron acceptor, and so it will take those electrons away because oxygen has a really high electron affinity. And so because of that, now this process can continue. As long as I'm breathing in oxygen, I can keep sending these electrons along the way. As the electrons move from pump to pump to pump, I am giving power to those pumps. Those pumps will pump protons into the inner membrane space. The number of protons inside will be higher than out in the matrix, so now they'll diffuse from high to low concentration. But they won't just diffuse, they'll follow their electrochemical gradient, which means they'll diffuse from high to low, chemical gradient, but they're also being kind of repelled, electrical gradient. We can actually measure voltage. Since charged particles are moving, we can measure the voltage to sort of like figure out how much of this proton movement is happening. We can also measure acidity, because a plus is a hydrogen ion. Remember, I erased it. But an H plus is a hydrogen ion. So as pluses are moving out in there, I'm changing the pH. The pH would be dropping if I keep adding a bunch of H pluses. Back to the Krebs cycle. So in the Krebs cycle, the whole point of the Krebs cycle was ripping stuff apart. I want the electrons. I don't care about the carbons. It's almost like the example I've given in class is if I have a book and all I really want out of it is the glue, then I steal the glue, and then I've just got like this pile of pages. Pretend that there were no threads, it was just glued. Now I've just got this pile of pages, so I'm just like, get rid of them. Same thing here with my pyruvate that I sent inside. All I want are the electrons, the chemical bonds. So when I steal the electrons, the chemical bonds, now I've just got these leftover carbon dioxides that I breathe out. Okay, this is all assuming that we have adequate oxygen, but it's possible that we're running low on oxygen. Maybe I'm sprinting, and so I can't get oxygen into my cells fast enough to keep up with everything. So if I'm sprinting, I'm doing this process. I'm going to keep doing this process of breathing, aerobic respiration. Aerobic means with oxygen. That's why we used to call it aerobics before they called it cardio. So I'll do this process, but pyruvate is piling up quicker than I can send it into the Krebs cycle. So because of that, we'll do an alternate pathway over here when I'm running low on oxygen. I call that anaerobic. In general, if you put a or an in front of a word, that means not that word. So if things are symmetrical, great. If they're not, then they are asymmetrical. And I put an in because there's already an A. Otherwise, I'd be like aerobic. So sort of like I don't say I have a apple, I have an apple. So anaerobic is without oxygen, and aerobic is with. So essentially what will happen is for this NADH to be able to move on inside, I have to be able to regenerate it. It has to be able to drop off electrons and pick up more. So in glycolysis, what will happen is eventually, whenever I'm backed up, this NADH will, instead of dropping off electrons here, it will stop dropping off electrons onto pyruvate. And then pyruvate will go through some chemical reactions and in us become lactic acid. If we were yeast, they'll become ethanol or ethyl alcohol. That's the type of alcohol that's in like beer and wine and whiskey and stuff. And the idea is this kind of works because lactic acid in us is what makes our muscles feel like kind of slow and fat and sluggish. That's why the first push-up is easier than the 10th and the 20th and the whatever. So lactic acid might be paralyzing our muscles. Remember, our muscles are made of protein. Proteins might kind of denature if they get too acidic, but is it possible that maybe in the presence of lactic acid, maybe nerves aren't firing correctly, or maybe my muscles are sort of like not contracting correctly because the pH is incorrect, it's too low, it's too acidic. So when I start sprinting, lactic acid will build up, and that's how I just feel like, and if it slows me down, now maybe my breathing can catch back up. So it sort of helps us out a little bit in a way, unless we're running for our life, then you know it's going to kill us. And then in yeast, like I said before, they make ethanol. So if I take yeast and grapes, throw them in a bottle, and I restrict oxygen by putting a cork in it, then they'll make wine. If instead I take yeast and I think it's corn or something like that, throw it in a bottle and ferment it, then that makes whiskey. And then, you know, if it's this, it's vodka. If it's this, then it's gin or whatever the different types of alcohol are. Last thing about cellular respiration is up here on this equation, you'll notice I have glucose plus 6 oxygens yields 6 CO2s, 6 waters, and 30-something ATP. So first of all, 666, how about that? But also, it's those carbons in the food that I ate, like I said before, are those carbons that I'm breathing out. Those oxygens right there are picking up electrons and becoming water. And then hopefully I net a whole bunch of ATP. That's the entire reason for the process. But cellular respiration is called respiration because I need to breathe in oxygen and breathe out CO2. Don't forget to look at those 2 free response that I mentioned at the very beginning. And thanks so much for listening. Next up will be photosynthesis. And I think that it'll be a little bit quicker just because once you understand cellular respiration well, photosynthesis is not too bad at all.